Welcome back to the stage, everyone, for our first round of company pitches. Following this round, there will be another networking break, so hold on to those conversations that you had started or that next person you're looking to find. Now I'd like to introduce to you the first four pitches of the day. First off will be Dr. Michael Weickert, who's CEO of Pacylex Pharmaceuticals. Michael's been developing drugs and startup companies in biotech and pharmaceuticals for 29 years. His experience and involvement in a number of companies is vast. He's driven oncology product development for at least 20 years. Recently, Pacylex has closed Series A, and as you would have seen in the news, and I will on their most recent discovery, and I'll let him tell you about their future plans and what's coming next. Dr. John Lewis is CEO of Entos Pharmaceuticals, an Edmonton-based biotech company developing genetic medicines using the Fusogenics drug delivery system. Dr. Lewis holds the Frank and Carla Sohonki Chair in Prostate Cancer Research, funded by the Alberta Cancer Foundation at the University of Alberta, and is a professor of oncology and chair of the Alberta Prostate Cancer Research Initiative. Don McCaffrey is president and CEO of Resverlogix Corp. His 38 years of corporate management experience included over 18 years in drug discovery and development. He co-founded Resverlogix in 2001 with Dr. Norman Wong, and his expertise in corporate operations has attracted an international team of experts to the company. The company's lead drug, apabetalone, RVX-208, has an 8-year lead on the market as the first epigenetic drug for the reduction of major adverse cardiac events. And last but not least, Mark Starrat is CEO of AazeinTx, a clinical stage biotechnology company dedicated to improving patients' lives with asthma. Mark has served on the board of governors at the University of Calgary and was vice chair of the investment committee and member of the executive committee. AazeinTx is developing an oral small molecule therapeutic that targets a novel neuromedicated airway reflex pathway to rapidly reverse asthmatic bronchoconstriction. AazeinTx is ready to begin a phase 2A clinical trial pending successful financing of $5 million Canadian. With that, I will turn it over to our first speaker, Dr. Michael Weickert.
Thank you very much. I really appreciate the opportunity to give you a brief update on Pacylex. As you just heard, we just completed a couple of weeks ago our Series A financing, which is critical to our next step. So please, next slide. So what is Pacylex? Pacylex is a company exploiting a new target in cancer, a process called myristoylation, which is a fatty acid modification of certain proteins. And this is catalyzed by one of two enzymes in the body, NMT1 or NMT2. And Luc Berthiaume at the University of Alberta discovered the important relationship between these myristoylation enzymes and cancer formation and progression. And that was recently published in Nature Communications last year. And then it was extended to solid tumors and breast cancer in a publication early this year. As part of that research, Dr. Berthiaume found the University of Dundee had a drug that was perfect for inhibiting these N-myrstyltransferase enzymes in the body and could be potentially used as a therapeutic. So we've in-licensed that family of drugs and are now developing a first-in-class drug targeting myristoylation. We have our first patient that we hope will be dosed by July, maybe even later this month. We have a pre-IND meeting that we've already completed with the US FDA. We have a Canadian CTA approved in March. We closed the Series A, as I mentioned, and should be in man in 3 sites in Canada, including the Cross Cancer Institute in Edmonton. Next slide. So we have an extensive team that has a lot of experience. Dr. Berthiaume, I just introduced. I've been introduced. John Mackey at the Cross Cancer Institute is our chief medical officer. He's run about 100 clinical studies in cancer, and he's the individual behind the clinical strategy. Ryan Height is yet another University of Alberta alum, who's our chief operating officer. We have outside board directors now. The company has added industry experts, Cindy Jacobs and Ajit Gill, and Mark Hewson is also on our board. And again, he's another University of Alberta individual, this time a professor of finance at the university. We also have a very experienced scientific advisory board, some of whom will be running clinical studies for us in the future. Next slide. We're tackling two very serious hematologic malignancies first. Diffuse large B-cell lymphoma and acute myeloid leukemia. Both have relatively poor 5-year survival rates. Initial targets will be in the relapsed and refractory patients, those who essentially have no options left. Next slide. And the reason why we're so excited about trying Pacylex-001 in these patients as soon as possible is because in all models that we've done, and there's 4 examples here of different non-Hodgkin's lymphomas and acute myeloid leukemia, what we see is that as you increase the drug concentration administered to the animals, you inhibit or you shrink the tumors. In most cases, you completely regress or eliminate the tumors in the animals so that they are undetectable when the animals are inspected later. So this is very exciting. It's always nice to see things that completely eliminate tumors. In some cases, like in AML, as few as 5 once-a-day oral doses was all that was required in order to completely eliminate the tumors at the higher doses of drug. Next slide. When we look at the profile in patients, we see if we look at these enzymes that I mentioned that are involved in myristoylation, the lower the expression of one of those 2 key enzymes, the worse the prognosis for the patients. So it does appear that this is a biologically relevant target and that this target is already affecting the cancer progress in patients. When you take cells out of patients and you treat them with our drug, you see that the cancer cells in the right panel are killed at much lower concentrations of drug than normal cells are affected by. Again, very promising that we could have a selective lethality for cancer cells, and that's certainly consistent with the in vitro results and the models that we've tested. Next slide. So this is a logical successor to a class of drugs that's well known in cancer called BTK inhibitors, that's Bruton tyrosine kinase. It's in a pathway that's essentially modified and regulated by the B-cell receptor complex. Pacylex-001 completely disrupts the B-cell receptor complex, so it has a much more profound effect on downstream effectors, which are generally oncogenes and growth and proliferation factors essential for cancer cell survival. So as a result, we have an efficacy that's 10 times greater or 10 times more potent than the known and commercial BTK inhibitors, dasatinib and ibrutinib, both of which are multi-billion-dollar selling drugs in cancer. So it's a great starting place for us to be an oral drug with 10 times the potency of very successful cancer drugs, targeting a space in a much more profound way. Next slide. And there's also very rapid development opportunities in both diffuse large B-cell lymphoma and acute myeloid leukemia, our first 2 hematologic malignancy targets. The 5 most recent approvals in these 2 indications are approvals that were done with less than 200 patients in open label studies. So you can very rapidly progress from the initial clinical investigation to registration and approval for these compounds in these indications. So we look forward to an accelerated development path. Next slide. And as you can imagine, success in a space that already has multi-billion-dollar selling products could be quite substantial. The sales model on the left just shows some projected sales for the product if we're clinically successful. On the right, you can see that Big Pharma has a very strong interest in BTK inhibitor products, and there's been a number of multi-billion-dollar exits in the last couple of years. Next slide. So just in summary, we have an oral once-a-day tumor-killing product that we believe is potentially synergistic with other therapies, has a unique mechanism of action, is first in class, has been validated scientifically, has been validated with investor capital in our one and only competitor that just came out in November, has long-term patent protection till at least 2041, and has the potential to be a successor to a very successful class of compounds. And we look forward to seeing it in the clinic and in patients in the next couple of days, or next couple of weeks. Thanks, everybody.
John, you're on mute.
My mic is on. Can you hear me? Are you able to hear me?
We can hear you now.
I just muted and unmuted.
Go ahead and start over, John.
Okay, we'll do. Are you able to hear me? Okay, I got a yes. Wonderful. I'm going to start presenting. Sorry, everybody. You think after a year in the pandemic, we get this sorted out. So I'm really pleased to be here today to spend a few minutes telling you about Entos Pharmaceuticals, where we're ushering in a new era of genetic medicines. Entos Pharmaceuticals is an Edmonton-based company with a platform technology to address a wide variety of genetic diseases. And so I'm the CEO, also professor at the University of Alberta. I co-founded the company with Roy Duncan, noted virologist at the Dalhousie University. We got a great CSO, Arun Raturi, with years of experience in formulation development, and a pillar of the community, Colin Koros, a serial entrepreneur, is heading up our business development. We're a very science-focused team. We got multiple PhDs, masters, et cetera, and the team has grown significantly over the past year. We have offices and labs both in Edmonton and San Diego, California. In Edmonton, we're in the Merck Invention Accelerator, and in San Diego, we're in the Johnson & Johnson J-Labs Accelerator. And we have extensive manufacturing capability in both countries, including a clinical manufacturing site in Edmonton, Alberta. And so at Entos, we're thinking big. We have a platform technology that can address a wide variety of genetic diseases, and we're really imagining a future world without any genetic diseases. So we really see a revolution that lies ahead, one that's really been laid bare by the pandemic and the success of genetic vaccines, for instance. And so, you know, this future really can include all diseases with the advent of safe, effective, and redosable genetic medicine technologies. And so, you know, the pandemic has really shown us that, you know, many of us already knew this, but I think now everybody's come to the realization that the age of genetic medicine is here. And really the key is the delivery platforms. We have the technologies to be able to increase or decrease gene targets. We have the ability now to edit genes. And really it's how do we get those tools into the body to make effective medicines. And really there are two ways of doing this, either with viruses. So in the gene therapy world, adeno-associated virus, and the vaccine world, adenovirus have been really successful. The challenge of anything that's virus-based is redosing. You can only really give a virus once, and then your own immune system produces a response against it. On the other hand, we have platforms called lipid nanoparticles, non-viral platforms, such as those by Moderna and Pfizer. And these, you know, really exciting, can be redosed, but they have challenges to deliver systemic or, you know, or whole body diseases. And so at Entos, you know, what we thought of, what if we could harness both the power of viruses and non-viral gene therapy in one platform? And was really enabled by this, you know, landmark discovery by co-founder Roy Duncan of this class of orthoreoviruses. And what they do is really interesting. When they infect cells, they cause all the cells around them to fuse together. And he realized immediately he discovered a novel viral fusion protein. And I'm showing it here beside the influenza fusion protein. You can see this is a very large multi-subunit protein that would be impossible to manufacture in a drug delivery system or genetic medicine platform. Roy, however, discovered a novel class of fusion proteins that he called the fusion-associated small transmembrane proteins. At Entos Pharmaceuticals, we've been working to incorporate this protein into a lipid nanoparticle-like platform that we've dubbed the proteolipid vehicle. And what distinguishes this platform from lipid nanoparticle mainly is the fact it can be used for systemic gene therapies, for systemic treatments that can deliver not just RNA, but siRNA to decrease gene products, DNA for complicated gene therapies and protein replacement approaches and vaccines, and of course, tools for gene editing and be able to actually edit genes that children are born with devastating childhood diseases. And we've worked to make this a safe, effective, and most importantly, redosable platform. We're partnered with, we have a number of fantastic manufacturing partners, including Precision Nanosystems in Vancouver, that allowed us to create a very scalable, single-step process for manufacturing that's now been used to make our first clinical batches of COVID-19 vaccines. So we can scale all the way from, a few microliters on the benchtop to hundreds of millions of doses. And even with our setup now with partner Alberta Cell Therapy Manufacturing, GMP facility in Alberta, we can currently produce over 400,000 vaccine doses per month in Edmonton, Alberta. And so this is a platform technology. We've been working since 2016 to establish more than 18 international partnerships with a variety of different companies worldwide in a variety of different areas, trying to find sort of the sweet spot for this gene delivery technology. I'm going to tell you, and so some of these have progressed to license agreements. We've been very successful in securing partners. We're very excited about the future of this technology in a wide variety of applications. I want to tell you a little bit about our COVID-19 vaccine. This is the project we started, just over a year ago that has brought the Fusogenix technology through into the clinic. We're currently preparing for phase 2 clinical trials of the vaccine. We decided to make a DNA vaccine using our platform because stability. We know the RNA vaccines work, but they have real challenges with stability, need to be kept at deep cold. DNA vaccines are stable in the fridge for over a year, effective, looks like with a single dose, produce what we think is really important for long-term durability and protection against variants, which is a T-cell response. And for this reason, we have a version that creates a pan-coronavirus response that will protect not only against future pandemics, but also emerging mutants. And of course, we have the scalable manufacturing that's really well suited to a pandemic situation. We have 2 candidates that we've selected for clinical development. VAX-001, similar to the approved vaccines, is an optimized full-length spike protein. This is now in the clinic, and again, we're getting ready to start phase 2. VAX-002 is this machine-learned, multi-epitope, pan-coronavirus candidate that we're very excited about, and we're thinking about this as the last booster you'll ever need. So I'm not going to show a lot of data, but I can summarize what we have so far. Really exciting data, both in animals and hopefully soon in humans. So both candidates produce significant antibody response. These antibodies were able to neutralize infection in neutralization assays. They stimulated strong killer T-cell responses, the right kind of T-cell response to effectively clear disease. We did a challenge study with VAX-001 in collaboration with Vito Intervac in Saskatchewan. We were able to protect animals from SARS-CoV-2 infection, importantly, either with a single or 2-dose regimen. And we also tested this in non-human primates, where we saw similar levels of neutralizing antibodies, whether we either gave a single or 2 doses of the vaccine. So very exciting. We're very optimistic that it can be used with a single dose. And we're currently testing this in a combination phase 1, phase 2, first-in-human clinical trial. Started at the Canadian Centre for Vaccinology with principal investigator and sort of leader of the field, Scott Halperin. In our phase 1, we'll be enrolling up to 72 participants at 2 dose levels. Once we establish safety and immunogenicity in phase 1, we'll move to phase 2, where we dose up to 500 participants, looking now at the effectiveness of either single or 2-dose regimens. As we move on to a phase 3, we'll be looking at VAX-001 and VAX-002, both as a primary immunization scheme, thinking mostly about the wide swath of the world that hasn't been immunized yet, and ideally as a dose or a booster, if you've already been dosed with one of the approved vaccines as the protection wanes. So looking forward, Entos has been very busy. We see, and as far as the COVID-19 pandemic is concerned, we don't see this as a foot race, but really we're in this for the long term. It's a marathon. We need up to 16 billion doses worldwide just for the primary immunization. We know that novel variants will continue to emerge, and it's likely to become endemic and require yearly boosters. We feel very strongly at Entos that it's essential that Canadians have access to a domestic supply of vaccines and boosters, and we're still in this. We're prepared. We have a platform that can service this need. More broadly, we see this platform really as a tool to go after any disease. We can now essentially write biological software to cure disease, and no disease is out of our reach. Thank you so much for your attention, and really appreciate being incorporated into this wonderful conference.
There we go. Please text me sooner if you can't hear me. That the mic shows on. Hopefully it's on. Okay, thank you. Next slide, please. It's our forward-looking statement. And I'd like to thank everybody for your participation today, ladies and gentlemen. It looks like there's 120 out there, so that's a real treat. So for those of you who don't know Resverlogix, at a glance, Resverlogix is a very unique company dealing in epigenetics. So we have a world lead in this category by quite a bit, 8 to 10 years. And it's given us some huge advantages. And with things like COVID showing up, there's some opportunity to use it during COVID. So apabetalone in 2020 was awarded the FDA's breakthrough therapy designation. So I'm sure you can imagine that safety profiles and efficacy were well reviewed there. We're very excited about where this is going. And we have safety. So this is a therapeutic that we will be utilizing going forward in COVID and other diseases. But we already have an extremely good safety profile. We have 4,200 man-years of treatment to date. And we've been working in patient groups such as cardiovascular disease, diabetes, chronic kidney, NALFD, vascular dementias, pulmonary arterial hypertension, and we will soon be starting in COVID-19. Next slide, please. So on April 6th, I believe it was, we were awarded the approval to start working on our COVID trials from Health Canada. That's a very exciting time for us. Next slide, please. And some of this was based on some of the work that's been done showing the effects on COVID potential with our dual mechanism approach, both dealing with ACE2 receptor reduction and cytokine storm calming. So we were published in Cell earlier this year, and it was a very, very extensive publication. The animal portion of it showed that induce the cytokine storm in the model, 75% of those mice will die. When treated with apabetalone, not a single mouse died. So that was a very good approach. Plus, on top of that, we had the cytokine storm calming, the ACE2 reduction. So we have a dual mechanism moving it forward. We should be into the first patients any day now. It's kind of a race between Edmonton, Montreal, and Toronto where the first patient will be, but we're getting fairly close. It's harder to get the trials started right now. As of December, there were 30 trials involving COVID, and just 6 short months later, we're looking at 900 global trials involving COVID. Next slide, please. So 6 months ago, we also were looking at US deaths of only 250,000 only. It's huge. Next slide, please. We've now surpassed 600,000. So when you consider that even today, the CDC announced a variant of concern, a new one, we think this is a very realistic approach for coronaviruses in general, all variants. Next slide, please. So we'll be moving this forward immediately, and the long-term potential of our dual mechanism should include all coronaviruses and their variants, as I mentioned. And we look forward to having this done within a year. And we don't feel we're late to the party here. Unfortunately, with these new variants showing up, it's difficult. Next slide, please. Now, in order to get this done, we're a small company like everybody else. We're only about 40, 50 people. So pretty hard to launch a global program like Moderna did. Moderna had a lot of money behind them. So what we've done is we've partnered with Eversana. This is for a complete commercialization. This is an extremely effective group moving forward. Next slide, please. They have 3,500 employees globally in all kinds of countries and fields. So this is going to enable us to launch this as fast as humanly possible. Next slide, please. Now, our lead program, we'll move to the next slide. Thank you. our lead program has the breakthrough therapy designation from the FDA, and we have been designing that and moving forward diligently. We will be trying to get this second half of 2021, now that some reduced time concerns of COVID and hospital and it's move this forward. Next slide, please. The data on this is pretty darn good. We were showing 63% relative risk reduction. This is pre-specified data with a p-value of 0.0002. This is why we got breakthrough therapy designation. Next slide. We were also able to show it in CKD patients. And again, with a very strong p-value and over 50% reduction in MACE events, MACE in this case being MIs, death, or congestive heart failure. So we're moving forward as fast as we can. Next slide, please. And the time frame on this is short. We do have approval from the FDA that an interim analysis time point, which would be about 18 months from now, can have an approval status. So based on our previous trial where we were at 67% relative risk reduction, and based on the approval rating here where we only need 22%, we are very confident that this will move forward and look forward to it. Thank you very much for your time, and talk to you soon.
Perfect. Thank you very much, Rob and your team at BioAlberta. We really appreciate the opportunity to be highlighted here today. And thank you for all the work that you're doing to enhance the ecosystem here in Alberta, much needed as we grow and expand the whole network across our province. My name is Mark Starrat, and I'm the CEO of AazeinTx, and we're undertaking a disruptive technology for treating acute asthma. And we've brought together a good team, highlighted by myself and my colleague Gord Anderson, handling the governance, the financial, and the operations side of things. But really, our management team is highlighted by my two other founders, Dr. Richard Wilson, on which his science is based. Patented pending science is based on this, over 35 years of neuroscience research, as well as our Chief Medical Officer, Dr. Lawrence Korngut, who is a neurologist and has done over 50 clinical trials himself, bringing that expertise to the table. In addition to that, we also have Janet Petrillo, who heads up our clinical development as the director of that. She herself has operated, created, and budgeted for more than 140 now clinical trials over the course of her career. And we're very happy to have as our lead principal investigator and advisor to the company, Dr. Gail Gavreau, who leads the Allergan Clinical Network based out of McMaster University and is world renowned. We're lucky that she happens to be in Canada, but is world renowned as having advanced some of the top asthma trials in the world over the last decade. So asthma may not be known in terms of the impact to everybody in the room, but it affects over 339 people, and surprisingly causes more than 417 deaths every year. And even more surprising is because of the unmet need, 30% of those deaths happen in an acute care center in a hospital, where all of the technology is available to treat these patients. There are over 11 million Americans that report at least one attack on asthma per year. And the market right now at about an $18 billion market that is growing by the end of 2027 to be a $26 billion market. And exciting for us is that we can navigate over half of the market through two regulatory authorities, both in Canada and the United States, representing 52% of the market. AazeinTx is developing a fast-acting oral treatment for acute asthma. And like I said, based on Dr. Richard Wilson's science, we've targeted a novel target involving the nervous system that has taken advantage of more than a decade of animal research that went into his Nature Communication publication that is one of the foundations for our intellectual property. And combined with that, we have a global exclusive license for all indications, and I'll talk about why that's important a little bit later, in Neo6860, a Neomed and Marne pharmaceutical compound that is a second generation best-in-class TRPV1 antagonist. The first generation of TRPV1 antagonists were very efficacious, but they had a side effect profile. It worked too well in a lot of cases. And so we're very fortunate that we have the best-in-class second generation compound that we're taking into clinical trials. And so it's a pill, an oral delivery mechanism, a rapid effect to deal with acute asthma, and has a specific action that I'll talk about in a second. And I'm not the scientific advisor. I invite you to reach out to us and talk to Dr. Richard Wilson, Dr. Lawrence Korngut, who can take you through the technical side of this. But really, Dr. Richard Wilson's research targets a neuronal pathway involving tiny internal blood sensors in the neck called the carotid bodies. And when activated by allergens, this neuronal pathway causes acute airway constriction and mucus production, the asthmatic attack, and contributes to long-term lung inflammation. And our technology acts by blocking this pathway, aborting asthmatic attacks and reducing long-term lung inflammations. As I talked about earlier, we have 3 patent families that are under our control. The first is the patent pending method, Dr. Richard Wilson's work. In addition to that, we have an approved patent, patent family 2 of Neo6860, approved in 18 countries. And just as important is an international PCT phase already has been positively reviewed that is going into in-country later next month that will extend the patent life. And that's a unique polymorph that is going to be able to take the Neo6860 compound and extend its life for a commercial capability. So we have an accelerated investment opportunity, 3 years from start to finish. We're very fortunate that all the great work in the compound of getting through phase 1, safety toxicology data, all of the time and money that it takes to advance. We're starting our program at the beginning of a phase 2A trial, about to go into first in-patient later this fall in November, subject to financing. And our program is basically a phase 2A clinical trial followed by a reformulation, and then about a year from November going into a phase 2B clinical trial. And at that stage, we'll turn it over for strategic opportunities. So a little bit of a history about our company. It was founded in the summer of last year between the 4 founders, and we got to work very quickly. First of all, in licensing the intellectual property from the University of Calgary, which the company now owns, and negotiating and executing on our global exclusive license with Admare. And we're in the middle of our phase 2A fundraise now, just starting last month, in preparation, as I talked a few seconds ago, about going into first in-patient in our phase 2A clinical trial, led by Dr. Gail Gravreau at McMaster, complemented by Dr. Richard Lee at the University of Calgary. And then we'll be going into, upon successful results, going into a phase 2B fundraise to take us through both the reformulation and the phase 2B trials. Subject to COVID restrictions in terms of the clinical trial, we feel very comfortable that we have a 3 years to exit opportunity for this strategy. So on AazeinTx, we're focused on 4 things. First and foremost is the creation, the operation, the budgeting of those clinical trials, something that we believe we have a team with a diverse and deep experience in order to be able to take advantage of this. In addition to expanding the IP portfolio, as they talked about, the method combined with our new polymorph compound, combined with our reformulated fast-acting formula to expand the intellectual property. And if Dr. Richard Wilson was beside me right now, he would be very excited about talking about all of the secondary indications that he is excited to use the compound to go back into the lab. And so it's interesting from we consider ourselves not really an asthma company, but a TRPV1 antagonist platform company, and believe very strongly that because of some of Richard Wilson's animal work and the extended work that we're doing right now, we believe that we have at the very least a risk mitigation strategy to our company, and at the very most a value creation strategy. In addition, at the end of that process, as I said, we're going to be looking at some strategic exits. It's interesting, the market itself is very monopolistic, dominated by two main partners in AstraZeneca and GSK. We don't consider them to be competitors. We feel very strongly that our pill has the ability to be very complementary to the existing inhaler market as a different way of getting therapies into the system to help and to address the unmet need in the acute asthma setting. But all of those companies on the right very clearly are opportunities for strategic acquirers or folks that we may want to work with in the future. And so, as I said, we're in the middle of raising a $5 million US dollar round right now, broken down in terms of clinical trial costs, budgeted down to the penny with a contingency. Happy to share that with you, as well as a very lean $1.3 million corporate budget over the course of the next year or so. So in summary, we have a fast-acting pill for acute asthmatic attack that complements the inhalers. The market is growing, $26 billion over the next few years. We believe we have a robust IP strategy. We're in the middle of our $5 million phase 2A clinical trial raise right now. And our plan of attack is 2 phase 2 trials, followed by a reformulation over the next 3 years before we look at strategic alternatives. And I would invite, again, thank you for the opportunity to present, and invite you to reach me out directly, and I can put you in touch with the scientific folks that would be more than happy to go through the details of our clinical development plan and our research. Thanks very much.
All right. Thanks, Mark, for your presentation, and thanks to Michael, John, and Dawn as well. We're just running slightly behind time. We're going to quickly transition you back to the networking. Find that person that you have to get their card before you go. Check out the tables where our presenters are at, and we will be back with our final 3 speakers in just a few moments.